In this video, let us see about the classification of enzymes. The enzymes are the proteins that made up of several polypeptide chains and they catalyze the chemical reactions. But not all the proteins are enzyme. For example, if we take the hemoglobin, they are just a transport protein. They doesn't involve in any chemical reactions. They just carries oxygen to all parts of the body. But when we take the structural proteins like myosin, they convert the chemical energy in the form of ATP to mechanical energy and the movement is generated. Let's see about the classification. The International Union of Biochemists classify enzymes into 6 types. They are oxidoreductases, transferases, hydrolases, lyases, ligases, and isomerases. The oxidoreductase enzyme catalyzes the oxidation and reduction reactions in which electrons are transferred from one form of molecule to the another. So, this group of enzymes usually utilizes NADH plus or NAD plus as cofactors. So, in this reaction, the hydrogen is transferred from ethanol to coenzyme NAD to form the NADH and the formation of acetaldehyde is happened here. There are 4 types of oxidoreductase enzyme. So, in this first one is dehydrogenases. So, it is the removal of hydrogen from the substrate. So, in this there are 2 types. First one is aerobic dehydrogenases. In this, the oxygen is the hydrogen acceptor. For example, xanthine oxidase. And another one is anaerobic dehydrogenases. This requires coenzymes like NAD, NADH and FAD. Example, pyruvate dehydrogenase. And the second type is oxidases. So they catalyze the removal of hydrogen from substrate to form water or hydrogen peroxide. Here oxygen is the hydrogen acceptor. Example, cytochrome oxidase. Third type is hydroperoxidases. So these enzymes use hydrogen peroxide or organic peroxide as substrate. Example, glutathione peroxidase. And the fourth type is oxygenases. So they catalyze direct transfer and incorporation of oxygen into substrate. Example, phenylalanine hydroxylase. Let's see about the enzyme transferases. So this enzyme catalyzes the transfer of a chemical group, that is the functional group, from one compound to another. In this reaction, the phosphate from ATP is transferred to hexose to form ADP and hexose 6-phosphate. Here the phosphate group is transferred from ATP to hexose. This reaction is catalyzed by enzyme hexokinase. The enzyme hydrolases. So they are the hydrolytic enzyme that catalyze the hydrolysis reaction by cleaving the bond and hydrolyzing it with water molecules. All the digestive enzymes are hydrolases enzyme. So let us see some hydrolysis enzyme. So first one is peptidases. So they hydrolyze the peptide bonds. Example, trypsin and pepsin. Next one is amylases. So amylases enzyme can hydrolyze the glycosidic bond of carbohydrates. Next one is endonucleases and exonucleases. So these enzymes hydrolyze the phosphodiester bond of nucleic acid. Fourth one is esterases and lipases. So these enzymes hydrolyze the ester bonds of lipids. Lyases enzyme. They are enzymes that catalyze the work by creating a double bond or adding a group to a double bond without involving hydrolysis and oxidation process. In this reaction, the fructose 1,6 bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. So this reaction is important in glycolysis process catalyzed by the aldolase enzyme. And aldolase is one of the lyases enzyme. So this aldolase enzyme is responsible for the cleavage of CC bond in this reaction. Isomerases. So this enzyme converts a chemical from one isomer to another. That is, it catalyzes the intramolecular structural rearrangement in a molecule. So in this reaction, we can see the phosphoglucomutase catalyzes the conversion of glucose on phosphate to glucose 6-phosphate. That is, the phosphate group is moved from one position to another in the same substance. And the last enzyme is the ligases. So this enzyme catalyzes the ligation or connecting of two big molecules by establishing a new chemical link between them. So the best example we all know that is the DNA ligase. So this catalyzes the formation of phosphodiester bond between the two DNA fragments. Thank you.